Hello students, as you are already aware from the previous video that I'll be starting the chapters for different groups like for the class 11, for the class 12 and for the NEET aspirants of 2020. Now I am taking a chapter of class 12 and that is the first chapter of class 12. It is basically for those students who are these days continuing at home as they are unable to attend the classes. The school classes are suspended. So for those students, it is very much important that they must continue their studies because the lagging which is taking place on these days that will finally affect the overall preparation for their board examination as well for the NEET preparation. So I am starting the biology of class 12. The first unit of biology is concerned with reproduction. In that we are having 4 chapters. The first chapter is the basic understanding of reproduction. The second is for reproduction in plants. The third is for reproduction in human. And the fourth chapter is for the reproductive health and all this contraceptive population control and all. So I start with the first chapter and that is the reproduction in living organisms. See, on the start of this chapter itself, first of all, we should try to understand the meaning of reproduction in scientific way. Reproduction in general, if you ask in general definition of reproduction, anyone can explain that it is the process for formation of new organism, it is the process for increasing the number of organisms. But if we come to the technical and biological definition of reproduction, it is slightly different from all these. So now we should come to the biological definition of reproduction. I am taking one example here. Suppose there is an amoeba. Second example I am taking of bacteria. Suppose it is a bacteria here. These amoeba and bacteria both were surviving in their respective medium, in their favorable condition. They were having plenty of water, organic food, everything. But all of a sudden, the environmental condition of this amoeba became unfavorable. The same happened for this bacteria also. Suppose both of them were living in the same pond and it happened to both of them. So in this unfavorable condition, what amoeba is doing, amoeba is undergoing cyst formation. In this cyst formation, we can easily observe that there is nucleus and this it is the cytoplasmic part and it is the exterior cover. So in this cyst formation, what this amoeba has done, it has released out all its water. It has made a tough cover, a dry condition, and that structure is here called as cyst. If we come to this bacteria also underwent, for example, in this condition, it has undergone endospore formation. Due to this unfavorable condition, suppose this bacteria has undergone endospore formation. So in this endospore formation also, what we have observed, all of its water and other organic chemicals, inorganic chemicals, mostly they are removed. But in both the condition, one important chemical they are preserving. This amoeba is working to conserve its DNA. This bacteria is also working to conserve its DNA. Here in this process of encystment or in this process of endospore formation, they have conserved their DNA. And this process we study in reproduction. Definitely in this condition, this case, we are not observing any increasing number of amoeba or any increasing number of bacteria. But still we are studying it in reproduction. It clearly shows that reproduction is not only multiplication, reproduction is not only increasing number of organisms, it is something else. What is that something else? In this condition, under all these unfavorable due to all these unfavorable factors, the amoeba is capable to preserve its DNA, and as we are aware, this DNA is the genetic material. The same is happening in this bacteria. It is also capable to preserve its DNA here. So that DNA is the genetic material. So we can simply say that this process of reproduction is working for maintenance of that genetic material. If we come to overall definition of reproduction, it is simply maintenance and continuity of germplasm. Germplasm nothing but genetic material. So it is maintenance and continuity of genetic material. Normally an organism tries to continue its genetic material. This continuity is normally taking place through formation of new young ones, but in certain condition, if it is not able to continue it, at least it will try to maintain it, and that maintenance we are observing here. So reproduction is basically maintenance and continuity of germplasm. It is nowhere related to increase in number. If number getting increased, that may be an additional part, but the main function is always to maintain and continue the genetic material. If we come to this, then we can easily understand the types of reproduction. If we come to types of reproduction, there are basically two types of reproduction. The one is said to be sexual reproduction and the second is said to be asexual reproduction. So if we come to these types of reproduction, sexual and asexual, then we can differentiate these two types on the basis of the basic definition of reproduction itself. If reproduction is the process for maintenance of germplasm, if that maintenance is done in its pure form, then it is a sexual reproduction, and if some changes has taken place, then we can call it sexual reproduction. What I want to explain, now listen carefully. In asexual reproduction, whatever genetic material is present in an organism, it is maintained as such. Means during this process, there should not be any change in genetic material. There should not be any new character, there should not be any new feature appearing in that particular genetic material. But the sexual reproduction simply means if during this process of reproduction, the genetic material has undergone some change. What is that change? That change is most commonly recombination of genes. So if a process of reproduction is involving recombination of gene, then we can call it sexual reproduction, and if no genetic recombination is taking place, then it is asexual. So the main point of concern is here no genetic change, and here in sexual reproduction there is genetic recombination. If we come to this genetic recombination, so at that level itself we can make some differences between the prokaryotic and eukaryotic living organisms. If we consider any eukaryotic, then you are aware that in case of eukaryotic organisms there is mitosis and meiosis type of cell divisions. In meiosis you have observed that there is prophase 1. In that prophase 1 you have observed that there is Pachytene stage. In that Pachytene stage you have observed that there is a process of crossing over, and this crossing over is the process which is working for genetic recombination. So from here what we understood here, that in case of eukaryotes, if we talk about sexual reproduction, that sexual reproduction must be involving meiosis, and in that meiosis there must be the crossing over, as this crossing over is the process for genetic recombination. So genetic recombination is the main characteristic of sexual reproduction. That is, in case of eukaryotes, if you define, the meiosis is the main characteristic, it is the main process which is representing that the sexual reproduction is taking place. Means for sexual reproduction in eukaryotes there must be meiosis, and that meiosis must be involving crossing over, as this crossing over is working for genetic recombination. But suppose if we take the example of prokaryotes, in prokaryotes there is no chromosome. As there is no chromosome, there is no any chance of mitosis and meiosis. Means meiosis cannot be taking place in a prokaryote. Then how can we define? Means then what should we conclude? Is there no sexual reproduction in prokaryotes? No, definitely there is sexual reproduction in prokaryotes as well. But the process will be different. It is not the crossing over, it is not the meiosis. Rather, there are different processes in prokaryotes for this genetic recombination, means that I am calling in simple word sexual reproduction. That sexual reproduction has to involve genetic recombination, and for that genetic recombination it has three different processes. The one is transformation, the second is conjugation, and the third is transduction. Means what I am concluding, for sexual reproduction in eukaryotes there must be crossing over, there must be meiosis. For sexual reproduction in prokaryotes there will be either of these three processes, like the transformation, conjugation and transduction. These are the three processes that can work for genetic recombination. So a bacteria can involve one or more of these three processes for sexual reproduction. That is the basic understanding of reproduction. In the next video, I will discuss about the reproduction or sexual reproduction in plant. Then I will come to sexual reproduction in plant. Thank you very much.